Some important question from respiration for Neet. Question 1. The site of glycolysis is options are cytoplasm, cytosol, intermembrane space of mitochondria, mitochondrial matrix, and mitochondrial inner membrane. The correct answer is the cytoplasm, or mainly cytosol, the soluble portion of the cytoplasm, which is outside the cell organelle. So glycolysis occur outside the mitochondria in the cytosol. Intermembrane space of mitochondria, this is the site for the oxidative decarboxylation. Okay, and mitochondrial matrix is for TCA cycle, and mitochondrial inner membrane is for the electron transport system. Question 2. In order for glucose to be used as an energy source, it is necessary that option 1, glucose be formed from fructose, 2, glucose be degraded to carbon dioxide, option 3, 2 molecule of ATP be invested in the system. This is the correct answer, that the beginning of the respiration, which is with the glycolysis. So glycolysis begin with the double phosphorylation in which 2 molecule of ATP are consumed. Here you can see that the glucose molecule first is phosphorylated. This is the first phosphorylation, and here ATP molecule is consumed. Then again fructose 6-phosphate is phosphorylated with the use of ATP molecules. This is the second phosphorylation. So this glycolysis begin with the double phosphorylation. Question number 3. Conversion of glucose to glucose 6-phosphate. The first irreversible reaction of the glycolysis is catalyzed by this reaction. The first phosphorylation is catalyzed by the enzyme hexokinase. So this is the answer. This phosphofructokinase carried out the second phosphorylation in which fructose 6-phosphate is phosphorylated to fructose biphosphate. Which one is the correct sequence of glycolysis? Here the important metabolites of glycolysis has been given. So the correct option is option number 2. Here glucose 6-phosphate is converted to 3-phosphoglyceraldehyde. Then 3-phosphoglyceraldehyde is converted into 3-phosphoglyceric acid, and then to phosphoenol pyruvate, and phosphoenol pyruvate is finally converted into pyruvic acid. Question number 5 and question number 6 are similar. Okay, question number 5. The intermediate compound common for aerobic and anaerobic respiration is citric acid, pyruvic acid, acetyl coenzyme A, and succinic acid. Here the common compound is the pyruvic acid. Question number 6 is, here the common phase between aerobic and anaerobic respiration is called common phase between the aerobic and anaerobic respiration is the glycolysis. Pyruvic acid is the end product of the glycolysis. Question number 7. Name the enzyme that convert pyruvic acid to ethyl alcohol. From pyruvic acid to ethyl alcohol, that is alcoholic fermentation, 2 enzymes are involved. One is carboxylase, another one is dehydrogenase. Here you can see the pyruvic acid in presence of pyruvate decarboxylase is converted into acetaldehyde. During this process, one molecule of carbon dioxide is released. And then the acetaldehyde is reduced to ethanol in presence of enzyme alcohol dehydrogenase. Question number 8. How many ATP molecules are obtained from fermentation of one molecule of glucose? From one molecule of glucose, by fermentation, the net gain is 2 molecules of ATP. During this process, 4 molecules of ATP are produced, but in the beginning of the process, 2 molecules of ATP are consumed. So net gain in fermentation is 2 ATP molecule. During the process of glycolysis, this NADH is produced, but after that, the NADH again consumed. Question 9. Yeast poison themselves to death when alcohol concentration reaches to dash in alcoholic fermentation. The correct option is number 3, that 13%. When the alcohol concentration is nearly 13% in the medium, it becomes poisonous for the yeast. Question number 10. In which one of the following process carbon dioxide is not released? The correct option is option 4, lactate fermentation, in which lactic acid is produced from pyruvic acid in presence of enzyme lactic acid dehydrogenase, and there is no formation of carbon dioxide. This lactic acid fermentation takes place in muscle cells of mammals and also carried out by the lactic acid bacteria.